case is this, that sometimes you have benign changes, normal polymorphism, and sometimes, unfortunately, you have mutation, which they are deleterious changes. So you cannot call them a normal polymorphism. So this is always a main issue, a very difficult, basically, decision to make. Specifically, when you find a new changes in human, you find a changes without previous report of that. So if you sequence human genome, for every human, you will see many positions which is different to the reference human genome that we have. And many of these changes are the first time that you see. It's not something that you see every day. Then it's very difficult to interpret that, especially when that person has some specific diseases without a definition. So you don't know if really this is a deleterious changes which is responsible for the disease or not. Many times we get patients, we get small babies, okay, with the genetic diseases, and the clinician writes for us that this baby has these different features. For example, it has hypotonia, developmental delay, seizure, but unfortunately, we do not have a specific diagnosis for him or for her. So, if it's possible, please do, for example, whole exome, check all the genes, and see which genes might be basically mutated. Okay, so you do that, so you sequence all the genes, but you end up with so many different changes. You check with the human genome reference, and then you find out thousands of changes in different genes. You go and check these genes. Some of these genes you don't know the function, so you cannot interpret what are these changes. Some of these genes you know the function, already know, and you look to the report. For example, you see that, okay, mutations in this gene cause seizure. Already there are papers which prove that. And then you focus, okay, so this gene prove, for example, mutation in this gene cause seizure and developmental delay, and it has been reported in many patients before, and now you have a changes in this gene. And this baby also has seizure, developmental delay, and these features. So maybe this mutation is responsible for the presentation that you see in this patient. Now you have to interpret, is this really this change responsible for that or no? And you look to this variation that you found, check to the databases, check to the papers. Okay, there is no report of this specific variation, because the variation in the gene may happen in every nucleotide. Let's say you have a gene which consists of 100,000 nucleotides. The mutation or polymorphism may happen in any of these mutations. Although you may have thousands patients before with mutations on that gene, but still you may get some new changes, some new patients with changes in the nucleotide, in the position on the genes that we never had before. And when you get to this scenario, always is difficult to interpret that if this is a mutation or if this is a polymorphism, normal polymorphism. All right. Okay. So when the sequencing of human genome basically made possible in terms of the cost, so you know that the first human genome sequencing was happened long ago. But do you know how long did it take to sequence the human genome?
10 years.
40?
14, I mean.
14? Close. Okay,
10 years.
13 years.
10 years.
13 years. One of these. So the Human Genome Project, the goal of Human Genome Project was to sequence the human genome, and it took 13 years to sequence the human genome. The first human genome which was sequenced. Around 50 to 60% of the sequence is only from one DNA, from one person. Okay, which tend to have a good DNA basically. Okay, and how much does it cost? Did it cost? 3 billion nucleotides, yes. Three, oh, okay.
Because this is an actual statistic.
Yes, you are correct. it cost $3 billion. Okay, so it took 13 years and it costed $3 billion to sequence one human genome. And do you know how much does it cost to do the sequence of whole human genome now?
But this is more important.
Okay, so depending on the devices that you use, it may cost below 1,000 USD, right? So it's possible to sequence the whole human genome with less than 1,000 USD and in a matter of several hours. So that is possible now. And this is obviously the progress will happen in the technology over the last 10-15 years. Okay? All right. So at the beginning, when obviously this was possible, many scientists, many centers started to sequence human genomes. They believe that probably after sequencing, for example, several hundreds of humans, then they probably find all polymorphism. And after that, when they sequence every human, then they won't have this problem that I explained to you, because they already basically sequence enough people to find all polymorphism. So you probably heard about 1,000 genome project.
Yeah.
So when they started this 1,000 genome project, they thought that, okay, so we sequence 1,000 genome and then after that we have all the polymorphism. But unfortunately it didn't happen. Okay, when they did this 1,000 basically genome sequencing, then they realized, no, this is not the case. Every human that they sequence has many, many variations that is not basically, is not possible to see them in other human or in previous sequences. Then, bigger project came in. For example, UK has 100K genome project, probably 100,000 genome project, to be able to define this polymorphism more accurately. But in fact, the base, my point, or the goal of basically my point is to say that although we sequence more and more humans, obviously we get to know more of this polymorphism. But in fact, we always have this problem. We always have this problem that when we sequence someone, we end up with some new sequences, some new changes, which is not present in the database and we haven't seen before. And we have the problem to interpret if this is a mutation, cause disease, or if it's a normal polymorphism. So what we have is this. Sometimes we have polymorphism, so they are normal, but unfortunately, sometimes these changes are deleterious, which interfere with the gene function. So on the basis of what I explained now, the polymorphism and all different forms of the alleles for the genes, you know that we can now talk about the genotype. So the genotype for every position that we talk might be homozygous. It means that we have two same allele, as we see here, or heterozygous. Two different allele and two homolog chromosomes that we have.
Oh, this is the X-Men.
Okay. So, any questions until now? I'm just talking, and I see that some of you prefer to get some rest instead of Any question? No question? Okay. So you probably knew already this.
Yes, Mr.
Okay. Anything new in this discussion? Did we learn anything, or it was just allele frequency? What else was new for you that you didn't know before?
Okay, the students who came to the first lecture, I think about 95% of what you said was already taught in the first lecture.
So why you don't basically stay?
Because most people were not in the first lecture. There was only like 20 students in the first lecture, but most of what you said was actually in the first lecture. It was already been taught.
So,
But the concepts were covered though. The concepts were covered, yeah.
okay.
Yeah. But it's
He
good, like
said
you
that
go in
this
more
was
detail.
totally new for him.
Yeah. But you went in more detail. So the concepts were covered, but it was more detail.
Okay. All right. Okay. So now let me define something else. So what is the meaning of recessive or dominant? So until now you learned about these, genotype values and everything. So now what is the meaning of recessive or dominant? Can you explain this? Listen please.
That is not expressed in the recessive, and the dominant one, the allele that expresses itself is the dominant one.
So it's fine, but let me just repeat what you said. Okay, recessive and dominant are the terms that used by Mendel himself.
Mendel, yeah.
Okay, and he used actually these terms, to define or to express the phenotype, not the allele, basically.
Phenotype.
So recessive and dominant are the definition that we use for the phenotype. We say this phenotype is recessive and this phenotype is dominant. So basically that's what is used for the character, for the phenotype.
But isn't that also based on the genotype? For example, recessive phenotype is based on recessive genotype as well.
Yes,
Yeah.
I'm just explaining. Yes, obviously it's based on the genotype.
I love it.
So because of that, sometimes we use the same term for the alleles. And we say this allele is recessive and this allele is dominant. But in fact, the definition is for the characteristic, for the feature, not for the alleles. Okay? And what is this definition? We call a phenotype recessive when it's present only when there is two alleles, okay, for that phenotype. So that phenotype called recessive. So it means if you need allele A in a specific position for that phenotype, you should have 2A. So it's only present in the homozygous state. So that phenotype called recessive. And the phenotype is called dominant when present in homozygous as well as heterozygous. For example, opposite A, if you have C as the dominant basically allele, so the phenotype related to C is called dominant when it's present with the CC genotype as well as AC genotype. But the phenotype related to A was only present when the genotype was AA. So that's called a recessive phenotype. Okay, so for the mutation in human, it's very important obviously to know this. Sometimes there are some mutations which create recessive phenotype or recessive diseases. And there are some mutations which create dominant phenotype or dominant basically diseases. Alright, so if you look to the function of the genes, there are differences among these mutations.